In today's video, we shall discuss about antimicrobial susceptibility testing. But before that, do subscribe to my YouTube channel and let me know your opinions and suggestions in the comment box. Antimicrobial susceptibility testing is a laboratory procedure used to determine the effectiveness of antibiotics or other antimicrobial agents against bacteria or other microorganisms. It helps healthcare professionals identify which antibiotics are likely to be most effective in treating a particular infection. In the current scenario, antimicrobial resistance has become a significant global health threat. Overuse and misuse of antibiotics have led to the emergence of bacteria that are resistant to multiple antibiotics, making infections harder and sometimes impossible to treat. AST plays a crucial role in combating this problem by guiding clinicians in selecting the most appropriate antibiotic treatment for patients. Before moving on to the details of AST, we shall first see what antimicrobial resistance, or AMR, is. Antimicrobial resistance refers to development of resistance to an antimicrobial drug by a microorganism. It can be of different types. Intrinsic or natural resistance refers to innate ability of a bacterium to resist a class of antimicrobial agents due to its inherent structural or functional characteristics. Acquired resistance is attained by the emergence of resistance in bacteria that are ordinarily susceptible by acquiring the gene's coding for resistance. Mutational resistance is the resistance developed due to mutation of the resident genes. Mycobacterium tuberculosis is a popular example. Transferable drug resistance is plasmid-coded transferred by conjugation or rarely transduction, transformation. Resistance-coded plasmid called our plasmid can carry multiple genes, each coding for resistance to one class of antibiotic. This is a tabular representation of natural resistance attained by different microorganisms to particular antibiotics. Moving on to the mechanisms of antimicrobial resistance. The mechanisms of AMR includes decreased permeability across the cell wall where the bacteria modify their cell membrane porin channels, either in their frequency, size, or selectivity. Prevents antimicrobials from entering into the cell. Observed in many gram-negative bacteria such as Pseudomonas, Enterobacter, and Klebsiella species against drugs such as imipenem, aminoglycosides, and quinolones. Another mechanism is with respect to the efflux pumps. The efflux pumps mediate expulsion of the drugs from the cell soon after their entry, thereby decreasing the intracellular accumulation of drugs. It is commonly observed in E. coli and other Enterobacteriaceae against tetracyclines and chloramphenicol. Staphylococci exhibit this mechanism against macrolides and streptogramins. Bioenzymatic inactivation is another mechanism. It is basically the inactivation of antimicrobial agents by producing various enzymes such as beta-lactamase enzyme production. Enzymes breaks down the beta-lactam rings, thereby inactivating the beta-lactam antibiotics. Another mechanism is by the bi-modification of the antimicrobial target sites within the bacteria. Commonly observed in MRSA, target site of penicillin, i.e. penicillin-binding protein, PBP, gets altered to PBP2A. Streptomycin resistance in Mycobacterium tuberculosis due to modification of ribosomal proteins or 16S rRNA. The factors contributing to AMR are overuse and misuse of antimicrobials, incomplete treatment courses, substandard infection prevention and control practices, limited access to clean water, sanitation, and hygiene, wash, globalization and travel, antibiotic pollution, lack of new antibiotics and diagnostics. Economic and policy factors. Moving on to antimicrobial susceptibility testing. So why do we do AST? Bacteria exhibit great strain variations in susceptibility to antimicrobial agents, hence it is necessary to determine the susceptibility of pathogenic bacteria isolated from the clinical specimens. Susceptibility testing of individual isolates is important with species that may possess acquired resistance mechanisms. Examples are members of the Enterobacteriaceae, Pseudomonas species, Staphylococcus species, Enterococcus species, and Streptococcus pneumoniae. A variety of laboratory methods can be used to measure the in vitro susceptibility of microbial pathogens to antimicrobial agents. Methods should be standardized based on international standards such as UCAST, CLSI, and ISO. Classification of AST methods. AST methods can be classified into disk diffusion methods, which include Kirby-Bauer disk diffusion method and Stokes disk diffusion method. Dilution tests, which includes broth dilution method and agar dilution method. The E-test, which employs a predefined gradient of antimicrobial concentration on a plastic strip. Automated methods that uses advanced technologies to streamline the process of determining AST, and finally molecular biology methods, which utilizes genetic and molecular techniques to detect specific genetic determinants of AMR. We shall now discuss each methods one by one. First method is the disk diffusion method. This method relies on the diffusion of antimicrobial agents from antibiotic discs into an agar medium, creating a concentration gradient. The diameter of the zone of inhibition around each disc reflects the susceptibility of the organism to the antibiotic. It is one of the most widely used methods. It is suitable for rapidly growing pathogenic bacteria, but unsuitable for slow-growing bacteria. Significance of Mueller-Hinton agar in disc diffusion. It is the best medium to use for routine susceptibility testing of non-fastidious bacteria for the following reasons. It shows acceptable batch-to-batch reproducibility for susceptibility testing. It supports satisfactory growth of most non-fastidious pathogens. It has minimal inhibitory effect on sulfonamide and trimethoprim. Hence, these antibiotics are better tested in MHA than any other media. Standard control strains. The following ATCC, American Type Culture Collection, strains are used as standard control strains, which include strains of E. coli, Pseudomonas aeruginosa, Staphylococcus aureus, and Enterococcus faecalis. Another important aspect of AST is the choice of antibiotic discs. Antibiotic discs, 6 mm diameter, are impregnated with standard quantity of antibiotic solution. Choice of antibiotic discs are done by the following criteria. Antibiotics should likely to be used for therapy of infections. Local prescribing habits of the antimicrobial agents. Resistant pattern of the locally prevalent pathogens. Cost, toxicity, pharmacokinetics, and spectrum of activity of an antimicrobial agent. First-line drugs. These are commonly used for treatment. Antibiotics that are currently being administered to the patient and the spectrum should be relevant to the organism isolated. Second-line drugs are restricted only to special circumstances. They are reserved for testing later if the organism is found to be resistant to all the first-line antibiotics tested before. Now we shall see the first technique in disk diffusion method. The Kirby-Bauer disk diffusion method. The Kirby-Bauer disk diffusion method is a test that determines how sensitive or resistant bacteria are to antimicrobial compounds. It's also known as KB testing. The steps associated with the procedure is inoculation, where a standardized inoculum of the test organism is spread evenly on the surface of a Mueller-Hinton agar plate using a sterile swab. After drying the surface of agar plate for 3 to 5 minutes, antibiotic discs are applied using either sterile forceps or multidisc dispenser. The plates are then incubated at 37 degrees Celsius for 16 to 18 hours. When tested for MRSA, results should be read only after 24 hours of incubation. The zone size is then measured using a ruler or vernier caliper. Finally, interpretation of zone size into sensitive, intermediate, or resistant is based on the standard zone size interpretation chart is done. Another popular technique is Stokes disk diffusion method. The Stokes disk diffusion method is a technique used to test the susceptibility of bacteria to antibiotics. It's a widely used method in clinical laboratories and is recommended by the World Health Organization. In this technique, MHA plate is divided into 3 parts. The test organism is inoculated on the central 1/3 and the control strain on upper and lower thirds of the plate. An uninoculated gap of 2 to 3 mm wide should separate the test and the control area on which the antibiotic discs are applied. Plates are then incubated at 37 degrees Celsius for 16 to 18 hours. These are some of the commonly used disc concentrations and interpretation of disk diffusion test. Next is the dilution method. In this method, serial dilutions of antimicrobial agents are prepared in broth media, and the test organism is exposed to these concentrations. The minimum inhibitory concentration, defined as the lowest concentration of antibiotic that inhibits visible growth, is determined. Dilution methods are of two types: broth dilution method and agar dilution method. In broth dilution method, serial dilutions of the antimicrobial agent is done in MH broth. Each tube is inoculated with a fixed amount of suspension of the test organism along with controls. MIC is determined by noting the lowest concentration of the drug, at which there is no visible growth. The minimum bactericidal concentration, MBC, can be obtained by subculturing from each tube, showing no growth, onto a nutrient agar plate without any antimicrobial agent. The tube containing the lowest concentration of the drug that fails to show growth, on subculture, is the MBC of the drug for that test strain. Broth dilution test can also be done using microtiter plates known as microbroth dilution method. The agar dilution method involves the incorporation of varying desired concentrations of the antimicrobial agent into an agar medium, molten agar medium, habitually using serial two-fold dilutions, followed by the inoculation of a defined microbial inoculum onto the agar plate surface. The lowest level that inhibits the visible growth of the organism is considered the MIC. Once an MIC is determined, clinically relevant breakpoints are applied to categorize the MIC. These breakpoints can be found on the FDA website, the CLSI M100 standard, the CLSI website, or the UCAST website. Moving on to e-test. The e-test employs a predefined gradient of antimicrobial concentration on a plastic strip. As the strip is placed on an agar plate inoculated with the test organism and incubated, the antimicrobial diffuses into the agar, creating a gradient. The MIC is read where the elliptical zone of inhibition intersects the strip. Automated antimicrobial susceptibility tests. Automated systems utilize various technologies, including turbidimetry, nephelometry, or fluorometry, to monitor microbial growth in the presence of antimicrobial agents. The systems interpret growth data and calculate MIC values. These systems offer several advantages, including increased efficiency, standardized testing procedures, and accurate interpretation of results. Most common types of automated systems are VITEC2 identification and antimicrobial sensitivity system, Phoenix system, and microscan walkaway system. We shall now see each of them in detail. The first system is VITEC2 identification and antimicrobial sensitivity system. VITEC2 utilizes a combination of fluorometry, turbidimetry, and colorimetry to monitor microbial growth in the presence of antimicrobial agents. The system measures changes in growth kinetics and generates growth curves to determine the minimum inhibitory concentration, MIC, of antibiotics. The system then monitors microbial growth in each well using optical sensors and generates growth curves. MIC is determined based on the growth curves. The system calculates the MIC for each antibiotic. The MIC results are interpreted by the system's software, which compares them to established breakpoints provided by organizations like the Clinical and Laboratory Standards Institute, CLSI, or the European Committee on Antimicrobial Susceptibility Testing, UCAST. Next is the microscan walkaway system. Microscan walkaway employs a combination of fluorometry and turbidimetry to measure microbial growth in the presence of antimicrobial agents. The system utilizes dry-form panels containing wells with antimicrobial agents, and the growth of microorganisms is detected optically. In this system, the test organism is inoculated into microscan panels containing wells with various antimicrobial agents. The panels are loaded into the walkaway instrument, which incubates them at the appropriate temperature and monitors microbial growth. The system detects changes in turbidity or fluorescence caused by microbial growth and generates growth curves. Based on the growth curves, the system calculates the MIC for each antibiotic. MIC results are interpreted using software algorithms that compare them to established breakpoints. Moving on to the Phoenix Automated Microbiology System. The Phoenix system utilizes both turbidimetry and fluorescence detection to monitor microbial growth in the presence of antimicrobial agents. The system employs dried panels containing wells with antimicrobial agents and growth media. The test organism is inoculated into Phoenix panels containing wells with various antimicrobial agents. The panels are placed into the Phoenix instrument, which incubates them and monitors microbial growth. The system detects changes in turbidity and fluorescence over time, indicating microbial growth. The system calculates the MIC for each antibiotic based on growth kinetics. MIC results are interpreted by the system's software, which compares them to established breakpoints. Now we shall discuss about the molecular biology techniques. Molecular biology methods for antimicrobial susceptibility testing, AST, utilize genetic and molecular techniques to detect specific genetic determinants of antimicrobial resistance in microorganisms. These methods offer advantages such as rapid turnaround time, high sensitivity, and the ability to detect resistance mechanisms directly from clinical specimens. Commonly used molecular biology techniques are PCR, multiplex PCR, DNA microarrays, next-generation sequencing, and loop-mediated isothermal amplification, LAMP. We shall see them one by one. PCR. PCR amplifies specific regions of microbial DNA that encode antimicrobial resistance genes or mutations associated with resistance. These resistance genes can include beta-lactamases, carbapenemases, and efflux pump genes. After amplification, the presence or absence of resistance genes is detected using gel electrophoresis or real-time PCR. PCR-based methods can provide rapid detection of resistance genes, enabling timely adjustment of antibiotic therapy. Multiplex PCR. Multiplex PCR allows for the simultaneous amplification of multiple target genes in a single reaction. This approach is particularly useful for detecting resistance genes associated with multi-drug-resistant organisms or specific resistance mechanisms, such as extended-spectrum beta-lactamases, ESBLs, or methicillin resistance in Staphylococcus aureus, MRSA. DNA microarrays. DNA microarrays are high-throughput platforms that allow for the simultaneous detection of multiple resistance genes in a single assay. Microarrays contain probes that hybridize to specific DNA sequences associated with antimicrobial resistance. After hybridization, the microarray is scanned to detect fluorescent signals, indicating the presence or absence of resistance genes. DNA microarrays are capable of detecting a wide range of resistance genes and mutations, making them valuable tools for surveillance and epidemiological studies. Next-generation sequencing. NGS technologies enable the rapid sequencing of entire microbial genomes or targeted regions of interest. Whole genome sequencing, WGS, can provide comprehensive information about antimicrobial resistance genes, mutations, and genetic relatedness among isolates. Targeted sequencing approaches, such as amplicon sequencing or targeted capture sequencing, focus on specific resistance determinants or genetic loci associated with resistance. NGS can facilitate the detection of known and novel resistance mechanisms, as well as the monitoring of resistance evolution over time. Loop-mediated isothermal amplification. LAMP is an isothermal amplification technique that amplifies DNA rapidly under constant temperature conditions. This method can detect specific genetic sequences associated with antimicrobial resistance in a highly sensitive manner. LAMP assays are relatively simple to perform and can be adapted for point-of-care testing in resource-limited settings. So how do we interpret antimicrobial susceptibility test results? AST methods measure the minimum inhibitory concentration of an antimicrobial agent required to inhibit the growth of the test organism. The MIC is the lowest concentration of the antimicrobial agent that prevents visible growth of the microorganism. MIC values are determined for each antimicrobial agent tested against the organism. These values are usually reported in micrograms per milliliter or dilution factors. Lower MIC values indicate greater susceptibility to the antimicrobial agent, while higher MIC values suggest reduced susceptibility or resistance. Interpretive criteria provided by CLSI, UCAST, or other relevant guidelines are used to interpret AST results. These criteria categorize microorganisms as susceptible, intermediate, or resistant to each antimicrobial agent based on their MIC values. Susceptibility breakpoints define the MIC values below which the organism is considered susceptible to the antimicrobial agent. Intermediate breakpoints indicate borderline susceptibility, while resistance breakpoints indicate resistance. Interpretive criteria may vary depending on factors such as the type of organism, the site of infection, and the clinical significance of the antimicrobial agent. Susceptibility categories based on interpretive criteria include susceptible, Microorganisms are inhibited by the usual systemic concentration of the antimicrobial agent, and therapy is likely to be clinically effective. Intermediate. Microorganisms show intermediate susceptibility, and clinical efficacy may be uncertain. Intermediate results may warrant higher doses of the antimicrobial agent or alternative treatment options. Resistant. Microorganisms are not inhibited by the usual systemic concentration of the antimicrobial agent, and therapy is unlikely to be clinically effective. Alternative antimicrobial agents are typically recommended for treatment. Some guidelines may also include categories such as susceptible dose-dependent (SDD) or non-susceptible (NS) to provide additional guidance for certain antimicrobial agent combinations. AST results should be interpreted in conjunction with clinical information, including the site of infection, patient factors, for example, immunocompromised status, severity of illness, and pharmacokinetic or pharmacodynamic considerations. Clinicians should consider the significance of the microorganism, the antimicrobial agent spectrum of activity, and the likelihood of treatment success when selecting appropriate therapy based on AST results. AST results should be accurately recorded and reported, including the organism's identification, the antimicrobial agents tested, their corresponding MIC values, and the interpretive categories, sensitive, intermediate, resistant. Results should be communicated promptly to healthcare providers to guide appropriate antibiotic therapy decisions and antimicrobial stewardship efforts. So, now we shall discuss about the importance of AST. According to estimates from the World Health Organization, AMR contributes to at least 7 LAKH deaths globally each year. Deaths caused by AMR are attributed to various factors, including ineffective antibiotic treatment, increased risk of infections with multi-drug-resistant organisms, and complications from resistant infections. So how is AST and AMR connected? By performing AST, microbiologists can determine which antibiotics are effective against a particular pathogen isolated from a patient specimen. Clinicians rely on AST data to choose antibiotics that are likely to be effective in treating infections while minimizing the risk of treatment failure due to antimicrobial resistance. Surveillance programs collect and analyze AST data from clinical isolates to track changes in resistance patterns over time, identify emerging resistance trends, and detect outbreaks of resistant infections. Before new antibiotics can be approved for clinical use, they undergo rigorous testing using AST methods to determine their activity against a broad range of bacterial strains, including those with known resistance mechanisms. AST is a cornerstone of antimicrobial stewardship programs aimed at promoting judicious antibiotic use, minimizing selective pressure for resistance, and preserving the effectiveness of antibiotics. AST data helps stewardship teams implement evidence-based prescribing guidelines, develop empiric treatment protocols, and educate healthcare providers about optimal antibiotic use practices.